Good. Okay, I'll jump right in. So I really do apologize that I am not there. I really wanted to be there and to meet all of the people in this room. I'm sure there's a lot of great people that deal with HABs, but we currently have an unprecedented freshwater cyanobacteria bloom that's moved into the western Mississippi Sound because of the Bonacary spillway opening in Louisiana. And it's very gnarly. I quickly threw some photos of this bloom into my presentation at the very end, so I'll show you. But like I said, it's unprecedented, and they just would not let me go. So I just 30 minutes ago received all of my samples, and I have 25 samples waiting for me. So I'll probably be here late, but that's okay. At least we know what's going on. Okay, so I'm going to jump right in. My name is Kristina Broussard. I think I heard Melissa introduce me, but I'm with the Mississippi Department of Marine Resources, and I have several roles here. I'm the biological program coordinator for the Shellfish Bureau, which means that I monitor oysters for harmful algal blooms and any other types of marine biotoxins. And also I am the lab director and the HAB task force coordinator. So when we had the bloom of 2015 of Corynea brevis, we developed a HAB lab and a HAB task force because it was so intense. Okay.
Say something, we might have lost sound.
Hello? Yeah. Can you hear me okay?
Yes, okay. I tried to move the speaker closer.
Yeah, and this is the tiniest little camera ever. Maybe that's better. Not the best lighting. Okay, so Mississippi in 2007, according to, or in coordination with FDA, developed the Marine Biotoxin Contingency Plan. And any states that work with oysters is required to have this contingency plan that states what we would do in the event of a marine biotoxin, including HABs. So, like I said, it was developed in 2007 and has gone through several revisions since then. The Marine Biotoxin Plan defines the DMR's harmful algal bloom monitoring program, which states that Mississippi will collect water samples twice per month during oyster season on harvestable reefs. Now, this is the bare minimum. We do a lot more than that, but that's what we are stating that we will do for sure. And that's just during our oyster season, which occurs in the colder months of October through April. I do try to continue that sampling throughout the entire year. We follow the NOAA Phytoplankton Monitoring Network's guidelines for sampling, which is where we use a 20 micrometer mesh and a 3-minute dredge tow to get a concentrated sample. We conduct qualitative analysis in-house, where we're just identifying any toxic phytoplankton that's present in those samples. If there's a bloom, then we will do a quantitative analysis, which is where we do a cell count, just like Alabama. I didn't quite hear his entire talk, but I believe they do cells per liter counts using a nuke chamber, and that's what we do as well. So these are the 4 locations that we collect twice a month. You can see to your left is the Western Mississippi Sound, 214 and 215B. That's at our northernmost and southernmost portions of our oyster reefs in the west. And then we also have Biloxi Bay to the right on the map. Maybe your left, one or the other. And that's 516 and 515. That's in our Biloxi Bay area where we have our Biloxi Bay oyster reef and our aquaculture oyster reefs. So additionally, we have trained our staff to conduct field observations when there's blooms possible. We also do flight overs for shellfish growing areas, and when we open shrimp season, we do this quite often. We have a member of our team that actually flies, and he will take coordinates of anything that he sees and get back to us. We can go by boat to that location. And then, of course, we investigate any reports of a possible toxic bloom. Would be any type of adjacent state agency, especially Alabama. I have a really good working relationship with Drew Sheehan at the Alabama Department of Health, and anytime there's an issue, he'll call me straight away, and we'll start sampling our East Coast and just see if it comes into our area, and also near Cat Island. So I think that's very important to maintain that working relationship with the different state federal agencies. And oddly enough, lately, Facebook has been our best friend. We've had a lot of fishermen reporting fish kills and blooms on Facebook and Facebook Messenger. So we will investigate anything that comes our way. So in the event of a marine biotoxin, we will close our oyster reefs immediately, and we will start doing cell counts. Now, as far as FDA is concerned and the model ordinance, Carenia brevis is the only phytoplankton, the only harmful algal bloom where there's a defined cells per liter count that closes an oyster reef, and that's at 5,000 cells per liter. This will trigger an immediate closure, and it has in the past. However, we do consider that number with other phytoplankton, just in an abundance of caution. Other toxic phytoplankton we will consider at 5,000. As we know, many phytoplankton don't become toxic until they reach the millions, 100,000. So literature is our best friend on that one. We'll also consult with FDA specialists in the FDA Marine Biotoxin Lab to determine if an oyster reef closure is necessary. So I just want to talk a little bit about NOAA's Phytoplankton Monitoring Network. I started working with PMN in 2009, and for the last 10 years I've been working with them. I started out as a volunteer because we were using Alabama to test our water samples for marine biotoxin, and it just got overwhelming for Alabama, and it was an easy task for me to take over. So I started as a volunteer with PMN, and I've moved into full-time HAB director over here. So eventually we'll be able to do our own ELISA test here. So we're building and growing, and it's been leaps and bounds since 2009. So I still use PMN's guidelines because they're just great. 20 micrometer mesh plankton net. Gives you a qualitative analysis to observe what's actually present. I look at live cells rather than preserved, like Alabama, and then if I need to preserve, I do have a batch of bluegulls available. Okay, so the history of HABs in Mississippi, there's been quite a few, but I'm just going to touch on the most important ones that we've seen in the past. The first, I'll start in 1996-97 when we had our first Carenia brevis bloom that we documented. Bloom occurred obviously in November through February, and at that time it was called Gymnodinium brevi. And there was a recall of oysters in the western Mississippi Sound due to this bloom. The highest cell count was near Cat Island, which is in the west, at 13 million cells per liter. There was a mouse bioassay conducted to open those reefs. And then in 2011, we had a Shetanella species bloom, July of 2011 in the summer months. It caused a very widespread fish kill. We did not monitor oyster reefs because Mississippi is closed for oyster harvesting during the summer months. That's the case we've made for the Vibrio vulnificus plan. And actually, this is the first year that we've been open for summer month harvest with aquaculture oyster reefs. And unfortunately, we got attacked by freshwater, so we've had a large mortality at our aquaculture sites. The Gulf Coast Research Laboratory in Ocean Springs, Mississippi did some cell counts for this Shetanella bloom, and NDEQ did some water quality analysis with FDA's Gulf Coast Research Lab. And FDA did some testing in August for Biotoxin, which they did not detect from this bloom. And then we have the biggest one that I've experienced, which was in 2015, 2016. And my friends in Florida from St. Petersburg called and said that it looked like this bloom was traveling north. Oh, I'm sorry, this is a different one. This was Carol Dorsey at that time was in Alabama, and Drew Sheehan was working with her. And hey, we've got some cell counts that are pretty high. You need to start looking. And so we did. And on December 1st of 2015, we saw the first sighting of Corynea brevis in our waters. And it lasted until January for 37 days. During that time, we conducted 165 cell counts in lab, and we did 98 of those were net toes. The others were whole water samples. We sent some samples over to Dauphin Island Sea Lab for an ELISA test to do a pre-screening before we did the mouse bioassay, and that came back good. And then in February, we did a mouse bioassay. Unfortunately, we had to send those to Florida, who was too busy, and had to send them to Brunswick, Maine. But we got it done. It was expensive, and we were able to reopen oyster reefs in February, which was pretty fast for us at the time. Now, I just want to make note that FDA has changed their beliefs in what, or not their beliefs, but their plan of action for detecting Biotoxin, and they now allow an ELISA test to be good enough to reopen oyster reefs. So I believe that St. Petersburg is training all Gulf states on how to do an ELISA test. I know that they're going to Alabama in this month, and we will be in St. Petersburg training next month, and then they're going to Texas the following month. So at some point, all Gulf states will be able to do an ELISA test to detect for Biotoxin without having to do a mouse bioassay, which is a very big deal, and we're super proud of that. And then we had a bloom in 2016. This was an Alexandrium monolatum bloom along with Serratium furca, a Kashiwo sanguinea, Gambardiscus species, and Dinophysis. It was amazing. This occurred from September to October. The highest cell count was Alexandrium at 1,818,000. The samples were sent to Florida Wildlife Research Institute and NOAA's PMN for a second opinion on if it was Alexandrium monolatum, the species, and it was of any toxins. So this is an aerial photo of the Corynea brevis bloom that occurred in 2015. You can see one of the barrier islands in the background, and you can see the striated red water. Beautiful photo. Another photo from the sky, you can see the red bloom. Again, this is Cat Island, I believe, and you can see the red water coming ashore. So this is a video. I'm hoping that it will play. It shows the movement of the 2015-2016 Corynea brevis bloom. And you can see when it comes into the area, and it comes in pretty heavy, and it explodes with the red and then moves out quite easily as well. And then it's on a loop, so it'll just replay. You can see the cell counts are in correlation with the NOAA's cell count chart. Our cell counts again in the west, central, and east. This is 2015-16 Corynea brevis bloom. They went above 1 million cells per liter in all samples collected across the entire Mississippi Sound for several days, and then they just slowly started dropping thereafter. Most notable water quality issue that we were having at that time was water temperature. The average water temperature is about 14, and you can see that it was 14 14 degrees Celsius, and you can see that it was really high for the majority of the time that we occurred. And the gaps in that chart are from Christmas and New Year's, when we chose not to. Okay, so during the bloom, during the 2015 bloom, we did a press release several times, but this is just one of the press releases that we did for beach closures, fish kills, and the birds and the marine mammals that were dying. We also looked at satellite imagery to determine the detection of a bloom in that area. You can see the Alabama coast is lit up in red from chlorophyll A. And that's it for my presentation, but I have a feeling you're going to have questions about some of the other things that occur in our state. So let me just spearhead those by saying that the Department of Marine Resources, we deal with marine activities. So we deal with oysters, shrimp, crabs, any kind of fisheries product. The Mississippi Department of Environmental Quality is what deals with more of a land-based and north of that area. They work with beach closures from fecal levels. They take E. coli testing. And unfortunately, my counterpart with DEQ, Emily Cotton, was going to travel with me today, but she too is immersed in a freshwater bloom, so she was unable to attend as well. So I had intended for her to talk a little bit about beach closures and testing for Vibrio and E. coli on the beaches, but unfortunately she's not here. I cannot talk on that topic. So just heads up when I ask questions. So before I end, I just wanted to show you some pictures of what we're working with right now.
No, this was.
That's what you wanted to see?
Yeah.
Okay.
Yeah.
All right, go ahead.
Everything okay?
Yeah, there was a question, but it's about what you're just moved into, so.
Okay, yeah, so I have just 2 more slides and they're all photos. So these are the pictures that we're working with now. It's a nasty green slimy bloom that moved into the Mississippi Sound from Louisiana waters. It looks like it just passed the Wrigley's and went into the Mississippi Sound yesterday, so it's just been a 24-hour bloom. It is a cyanobacteria called Dolichosporum, which is formerly known as Anabina. It does produce saxitoxins, which are toxic for humans and animals. It's freshwater associated, and you can see the photos. So this was yesterday's boat trip for us. We actually found it yesterday. The water sample to the left is a whole water sample that is not a net concentration sample. Pulling my net to try to get a concentrated sample, but it got a little clogged. And then the wake of the boat, you can see to your right, it's green, just the whole wake of it. So that's the environmental pictures. Then these are the microscope pictures. So it's Dolichosporum is the name of the freshwater phytoplankton that's moved in. You can see that it has these circular clear bubbles throughout the chain. Those are those are heterocysts. The aconites are a little bit bigger, and those are what cause it to become buoyant and float at the top. And then when the cells lyse, they release a lot more toxin. They actually have 4 different toxins, but the one that we're concerned about now is saxitoxin.